Hello everyone. Today we will see epistasis. Basically, it is an interaction between two or more genes to control a single phenotype. Example of epistasis is a coat color in mice. In mice, three varieties are there phenotypically depending upon what is its coat color. It may be gray or agouti coat color. Second, albino or white. And third, the mice will have a black, exact dark black coat color of their hairs. These varieties are possible because of the interactions which are taking place between the genes. These interactions are having two effects: one, epistatic, and second, hypostatic. The epistatic allele is going to mask the expression of another allele, like it is having a dominating effect, and the hypostatic is the one whose expression is masked. So these interactions are several, but the one which we are going to study today is a recessive epistasis, where recessive allele is going to mask the expression of dominant allele located on some non-allelic gene. Here, if you see on mice coat color, the wild type, the one which is predominant, is grayish coat color. Although it is looking gray to us, but if you see the pattern of hair of a gray or agouti mice, it is having an alternate coloration of black and yellow. So these patches, alternate bands of yellow and black, will give a grayish appearance, which is called as agouti pattern. Second one is a white or albino, and third is a black coat color mice. So if we want to demonstrate how these three varieties of mice are produced because of the interaction of gene, we have to show a cross between true breeding species, the one which is producing the agouti coat color mice generations after generation, is crossed with the another variety which is producing a white coat color generation after generation, and we will check how the next generation F1 and F2 is produced. So in F2 we are expecting the 9s to 3s to 3s to 1 ratio is modified to 9s to 3s to 4. How it is modified? That is because of the recessive epistasis, where the recessive allele, that is CC, in homozygous recessive condition is going to mask the expression of another non-allelic dominant allele, that is capital A, and final result is albino mice. So although A, which is a dominant allele, ideally it should expressed, but as it is an example of recessive allele, small c will not allow expression of capital A, and the result will be albino mice. So here the ratio will be modified to 9 as to 3 as to 4, that is 9 agouti mice, grey color mice, as to 3 black mice, as to 4 white or albino mice. If we see a cross, means a true breeding species which are producing a agouti coat color, genotype will be obviously capital A, capital A, capital C, crossed with a albino mice which is true breeding, genotype is small c. They are going to produce haploid gametes respectively. Fusion of gametes will give us F1 generation. Next, crossing of F1 with F1 will give 16 possible offsprings. How we will determine that what is a genotype of these offsprings? So we need to locate first for finding 9 agouti pattern. Focus on A. Where in your genotype, wherever capital A is there and capital C is there, that coat color will give agouti mice. Next, the focus will be on another combination, means how albino and black combinations are produced. So in case of black, capital A has to be absent. So whenever capital A is there and capital C is there, we are going to get a agouti coat color mice. But black combinations are also possible. in 3 number where capital A is absent and capital C is there either in homozygous dominant or in heterozygous dominant condition. So in homozygous dominant, it means it may be capital C or capital C small c. Likewise, there are 3 genotypes and that is going to give phenotype black. 3 remaining, four remaining, which are albinos, where small c is there in homozygous recessive condition. That is actually a epistasis where recessive allele are going to mask the expression of dominant allele which is located on non-allelic pair. That is, small c is going to mask the expression of capital A and the result is albino. So what is our final result? 9 as to 3 as to 3 as to 1 ratio ideal if genes or pair of alleles are following Mendelian principle, we are expecting it. But here it is a modification interaction where the ratio is modified to 9 as to 3 as to 4. I hope you have cleared a concept of recessive epistasis. Thank you so much.